Hello and welcome back to Tamali's classes. Today we'll talk about organogenesis and vulva formation in Caenorhabditis elegans, also known as C. elegans. So let's start. So this C. elegans was first discovered by Emily Moppus in 1897, and also it was the first organism whose genome was completely sequenced. This organism has a transparent body. It's very small, okay, and the life cycle is also very short. This is a multi-organ and eukaryotic organisms, and also this the life cycle of the C. elegans, it has a 4 larval stages: L1, L2, L3, and L4. Fine. So this C. elegans is very easily can be maintained in a solid or liquid medium along with non-pathogenic bacteria known as E. coli OP50 as a food source. And also, since it's transparent in nature, researchers find it easy to work with because they can visualize everything that is occurring inside the body. So this C. elegans, they have two natural sexes. One is hermaphrodite with XX and male XO. So this hermaphrodite, that can produce approximately 300 progenies, okay, by self-fertilization. Don't you think that's amazing out here? So they also allow easy generation of genetically identical progeny. This C. elegans, this genes of C. elegans, that can be easily manipulated using different genetic tools such as CRISPR-Cas9 gene editing and also genome scale RNA interference. Now let's move on. So now this is the structure of C. elegans. If you see out here, the C. elegans structure, in the center you can see. So this has been taken by taken from the one of the research paper. So see, you can see the structure out in the middle. See, this is the vulva out here. This is a small pore opening, okay, through which eggs can be released. The zygote can be released. The newly formed organisms. So this is the embryo part out here. This is the oocytes, spermatotheca, the male part, the female part out here. Okay, this is the gonad region, and this is the main part out here. Okay, so this is very important. Vulva development is very important. If the vulva is not developed properly, what will happen? There will be no fertilization of sperms and eggs out here, and there will be no formation of new organisms. Okay, properly they will not come into, they have to come together in this area. They have to come here, the sperm has to come here, the egg has to come here, and they have to, then they'll fertilize. So if vulva is not developed properly, this cannot occur. Okay, now see here, this is the hermaphrodite tail, and this is the both male and female. Okay, and this is only the male part. So you can see the tail, how it's differing between the male and the hermaphrodite part. Now moving on to the other, this is the macroscopic image. You can see through the microscope, it looks like this. Okay, so the fats out here is mainly stored in the intestinal and epidermal part of this C. elegans. Now see, this is a more and you can see this diagram is a bit more clear than the previous one out here. So you can see this is a vulva out here. You can see this opening out here. This is a vulva out here. The intestine part, this is a spermatotheca, and the fertilized egg is forming, and then it's being released. Okay. Now development of vulva. Now before I go to this part, I'll just say you a bit about the development in a more easier way. So out here, I told you that vulva is necessary. Why? Because for copulation and also for this egg laying. Okay, the important ones I'll tell you. They say for egg laying and also for the copulation with males. Okay, so this vulva will develop in which stage? During the post-embryonic development stage. Okay, and it will connect the developing uterus with the external environment. I showed you that there's a pore opening type structure. So this is how they will connect with the external environment. Now coming back again, since adult vulva, they will comprise of 22 cells, which are progeny of 3 ancestral cells. I'll show you. Okay, then vulva also serves as a paradigm for organogenesis. It's also an example for tissue remodeling. So not required. So okay, so moving on. This G protein coupled receptor, known as named as SR13, will act negatively on the vulval development via G protein GPA5. So that means it will not help in the development of vulva. Okay, by which protein? By G protein GPA5. Remember, and who is the G protein coupled receptor? SR13. So this one you should remember. This is the important one. Now moving on, I'll show you this part, the 22 cells wala part. I'll tell you, okay, what it is. So mat daro, isko dekh ke mat darna, don't get scared. So now first I'll just show you the basic part. This is the anchor cell out here. Okay, this is the anchor cell, and these ones, see, these ones are the vulval precursor cells, or also known as VPCs. See, P3P, P4P, P5P. These are all in the form of P and P form. Okay, so P3 to P8 is that, and then This is the number 8 and this is the P form. Okay, so these are all these vulvar precursor cells and they are going and attaching with the this epidermal layer HYP7 epidermis. Okay, now out here only this P5, P6 and P7 they will be acting and the other ones will not be acting out here. I'll tell you why. Now coming back to this part, see, follow this very properly. Okay, look at this and understand the process. Okay, see, the vulval precursor cells, I told you there are 22 number of adults are there. Now among them there are 6 numbers which are also known as equivalence group. I showed you from P3 to P3P to P8P. Okay, so they will form the vulva part. Now this gonad cells, they will form this VPC cells with the help of anchor cells. Okay, now anchor cells, there will be some inductive signals. You have seen in the previous diagram out here, there are some, you can see out here, there are some inductive signals in this green part, inductive signal out here. Okay, so there will be some inductive signals which will help control the development of the equivalence group. Okay, we have seen out here the anchor cells is there are some signals being received by the anchor cells and they are leading to formation of this P3 to P8P vulvar precursor cells. Okay, the equivalence group. So now what are those signals that are involved out here? Lin3 protein. This protein will help generate those inductive signals which will help in this process. Okay, so this will also help in this Lin3 protein. What does it do? It acts as a growth factor. It is necessary for induction of C. elegans vulva by the anchor cells. Okay, now this, how does Lin3 protein help? You know, the Lin3 protein's concentration, depending on the concentration of the Lin3 protein, it can lead to formation of either the vulva cell or the hypodermal cell. Okay, so remember, depending on the concentration of the Lin3 proteins. Now Lin3 protein, what does they do? They just act similar to the EGF, okay, epidermal growth factor. Just they will act similar to, they're acting like a homologous homolog to this EGF. Okay, so now let's see the functioning of Lin3 protein. Now I told you that based on the concentration of Lin3 protein, the functioning will occur. So if Lin3 protein is highest, okay, so concentration is highest, it will lead to primary precursor cells. If it is less concentration, it will lead to secondary precursor cells, and if it's negligible, then tertiary precursor cells. So this is not so important out here. We'll talk only about the primary and the secondary precursor cells. So primary precursor cells, they will lead to the formation of P6P, remember which I told you in the previous slide, P6P, P7, P8, all those things. So P6P will be formed by first precursor, primary precursor cells, and P5P and P7P will be formed by the secondary precursor cells. So when P6P will divide, when there is a cell division occurring, they will form 8 vulval cells. Remember this, okay? 8 vulval cells. And P5P will form 7 vulval cells, and P7P will be forming 7 cells. So this is all happening with the help of the differentiation pathway, remember. Okay, so differentiation pathway, they're occurring 8, 7, 7. Total, if you do, you will see that there are 22 vulval cells. So which is also the number of cells present in the adult, or you can say vulva. Okay, so now vulva is important so that this all development occurs in the correct place. Okay, or else what will happen? The gonad will not receive the sperm and egg and there will be no fertilization. I told you that before already. So now moving on to the steps of vulva development. There are 5 steps. Okay, don't get scared. I'll make it easy for you. So see, this diagram is again taken by another research paper, okay, by Paul W. and Sternberg. So now coming here, you can see this part out there. The first, there are again 5 steps out here. I've marked it as 1, 2, 3, 4, 5. You can see here, and also in the 4th part, okay, in the 5th part, we'll be seeing there are 2 again, A and B. Okay, so now let's see the first part out over the first one. It's very easy. I've already explained to you before this one. So in the first part is this generation of VPCs or vulval precursor cells. Okay, this is the first part. So we know that during the L1 and L2 stages, 6 vulval precursor cells are formed from 11 PNP cells, okay, in the ventral epidermis. So this is, we have already learned this one. Also second part, we have seen this VPC specification. We have already seen how the patterning is occurring, how primary and secondary is being formed. So that is coming under the second step. Okay, the third step is the fate execution, primary fate execution, secondary fate execution, and third fate execution, or the tertiary fate execution, or it's also known as the generation of adult cells. So here you can see that 22 cells were present in adult cells. So there are again of 7 types, VULA to VULF. Okay, so there are different types out here. So they will all differ in the pattern of gene expression and also in their cell biology. So Remember the 22 cells that we have seen, the 22 nuclei that we have seen, they are of 7 types from Vul A to Vul F. Okay, so this is the fate execution. Fine. Now here, if you see the diagram out here, there is one UV1 induction out here. So here we have seen out here, this UV1, I'll explain to you, don't worry about that. So this is the third part. Then moving on to the fourth part, where ankle cell invasion. This is the fourth part out here. So what's happening out here? Lateral signal is taking place. Okay. So the ankle cell out here, it will extend the process to the center of Vul F. We have seen Vul F is another one of the 7 types. So it will extend its process to the center of Vul F, and this Vul F will form a hole in the epidermis. We have seen in our previous slides that there is a hole in the epidermis, okay, which will we have seen here, right? See this one out here. You can see this is out here forming this, okay, linking with the epidermis part. Again coming here, same thing it's saying out here that it will form a hole in the epidermis, okay. So if you see fourth part out here, see this ankle cell out here. Let's show you with my diagram. See here, this ankle cell out here, this ankle cell, okay, this ankle cell out here, see, is invading into the vulval morphogenesis part. This is the fourth part out here, okay, ankle cell invasion. This is the ankle cell invasion in the diagram if you see that. So this ankle cell is coming and it is entering into the VULF part and is forming a hole in the epidermis. This is this AC invasion, okay, fourth part. Now moving on to the 5 part. So 5 part is having again 2 types, okay. So 5 is the morphogenesis of vulva. That means morphogenesis means developing of the morphogenetic character parts and all more, okay. So the 7 type of vulval cells, what they will do, they will invaginate and they will form 7 distinct toroids, okay, connecting to the uterus, and also the hermaphrodite will mold to adulthood. Now don't be confused, look at the diagram out here, okay. So there are again 2 parts in the 5th part, the morphogenesis part of vulva. See, we have all vulva development, all steps have been done. At the end is the morphogenesis of vulva, okay, character formation. So see, so there are two sets out here. First is, see, this one we have seen ankle cell invasion we have seen. So in ankle cells again there are, during the end of L2 stage, we saw that in L1 and L2 stage the things were happening. Now generation of anchor cells occurs at the end of the L2 stage. Remember, so in morphogenesis part there are 2 things happening. One is the generation of the anchor cell, and number 2 is the uterine patterning, okay, uterine patterning, okay. So first we'll talk about the generation of anchor cells. In the first part, if you see, it's happening in the end of the L2 stage, okay. Now come to the second part, uterine patterning. What is happening out there? So there are 3 ventral uterine precursor cells, okay. So they will form 12 grand progeny, and among them the 12 grand progeny again divided into 6 part, different part. So 6 will be induced by the anchor cells forming 6 pi cells, okay. The other 6 is remaining out here, okay. So this coming to the left hand part, so you can see out of this 6 induced by anchor cells, they'll be forming 6 pi cells, okay, and this will lead to division of cells, okay, and forming 4 progeny. Remember, this 6 pi cells, they will divide and they will form 4 and 2, okay. 4 will be this side, 2 will there. I'm just separated to show you the difference. So the 6 pi cells will divide and they will form 4 progeny, which are, these 4 progeny are induced by the VULF cells, okay, of the vulva primary lineage to become UV1 cells, okay. Remember, यहाँ पे क्या होता है, यहाँ पे in this part ना there are 2 type of cells being formed, UV1 and UTSE cells, okay, UTSE, UTSE cells and UV1 cells, okay. So UV1 cells out here are formed by this 4 progeny. Remember, 6 pi cells है, the 6 pi cells are dividing and they're forming 4 and 2. So the 4 progeny are induced by VULF cells to produce UV1 cells. The 2 which are remaining, they will bind with the previous 6 cells to form 8 cells, and they will fuse to form the UTSE cells, which will again fuse with the anchor cell, okay. They will again fuse with the anchor cell. Now this out here is forming UV1 cells. They will UV1 cells will again fuse with the VULF cells. The coming back, see out here. Now see the diagram if you have understood. Now you can see the diagram out here. See, pi cells हुआ था, okay, there were 6 pi cells formation, and also there were anchor cell. So we have seen that there were, how many pi cells were there? We saw that there are 6 pi cells, okay. They're leading to formation of 2 type of cells, UTSC and UV1, and we saw at the end that UTSC was binding to the anchor cells. See, देखो यहाँ पे anchor cells are binding with the UTSC, okay, and also UV1 is being formed at the end, Okay, so this is the step. Okay, this you can see even induction out here. So ये है step. So finally ये होता है, and again both are this UV1 will bind with the VULF, okay, and UTC will bind with the anchor cells. So ये VULF और ये anchor cells. So this is how the vulva is developing. Okay, so this I've just shown you out here how they are generated. So you can see that there are 11 PNP cells. I've already told you they are found in the L1 larval stage. So this development of vulva depends on the generation of PNP cells. Now, very important, there are Hox genes present. There are 6 number of Hox genes present in C. elegans. Remember this, okay? Which are responsible for specifications of VPC, that is P3 to P8. So, कौन responsible है? Hox genes. There are 6 number of Hox genes. So, what are the Hox genes? Anterior CEH13, central LIN39, MEB5, posterior group genes. There are 3 out here, EGL5, PHP3 and NOB1. So, these are all the 6 number of Hox genes. They play a role in the early patterning events of nervous system. Okay. Now, how is this regulated? Very, very again important. We have seen that LIN39 will send the signals and all. Remember. So, how is LIN39 regulated? Very important part. See, LIN39 can be inhibited by MAB5. Okay, or it can be positively regulated by WNT and LET23 signaling pathways, but it is inhibited by the MAB5. Remember. Now, LIN3 will inhibit the EGF1 expression. Okay, this will lead to inhibition of the fusion with HYP7. Remember, epidermal cells. It will inhibit that part. Okay, it will inhibit the attachment fusion. Also, it will regulate the proliferation and the responsiveness to LIN3. Okay. Now, remember this LIN39 function is very, very important for regulation of vulvar development. Okay. So, LIN39 is inhibited. It is positively regulated. It itself inhibits and it also regulates. Okay, there are 4 ways how it can regulate the vulvar development. Now, coming back, if you see the down part out here, I've just mentioned some important topics, important parts out here. WNT signaling pathway, we saw that it was regulating. So, how does it regulate? It is essential via the BAR1 beta catenin because it maintains the LIN39 expression. Okay, in P3 to P8 P cells. Also in this, if you say cell cycle, the cyclin E, okay, it will help in the proliferation of VPCs. LIN4, LIN14, LIN28, they will control the G1 to S transition of VPCs in the cell cycle. Okay, so it will help in controlling the G1 to S transition of VPCs. Also, CKI out here, 1 is the cell cycle inhibitor. Remember, it will inactivate, inactivation will result in the VPC division and it will block the mitosis. So remember this is the cell cycle inhibitor. They will help in the transition process, proliferation of VPCs, and also they will help in maintaining the LIN39 expression. So these are all the things that are regulating the vulva development. Now there are some 3 major signaling pathways which will help in the patterning of the vulva and uterus. So you can see out here, just for your knowledge, I've given this. Okay, so ligands these are all the ligands. These are all the receptors out here. These are the signal transduction. What is happening out here? This is the regulators and these are the target genes. Okay, so you have to remember this EGF signaling pathway. You can say notch signaling is also there. Then you can say WNT signaling. Remember I told you about BAR1. How does it help? So this one is out here. PRY1 is inhibiting. See, BAR1 is helping regulating the expression and PRY1 is inhibiting the action of, or you can say it's inhibiting the expression of BAR1. Okay, so the red ones are inhibitors and the green ones are regulators. Okay, so this is all the WNT signaling by P7P polarity and this is by the VPC competence. So you can see this pathway. Okay, see out here, ये भी देखो, the LIN3 pathway is very important. Receptor ये है, transduction हो रहा है. Regulator कौन है? LIN31 and LIN1 out here. And the target genes are LIN39. Okay, so this is for your knowledge. तो मैंने यहाँ पे बस थोड़ा और बताया है कि सेम पहले वाला ही चीज है. These are all the previous ones only, nothing new. So see how this is how they are all dividing. These are rounds of division out here. Okay, so you can see these things. So this is primary, secondary, and tertiary. So we know that primary and secondary में हो रहा है. And this tertiary, it's like tertiary. You can see this tertiary fate is non-vulval. Okay, and there's only one round of division. One round of division out. You can see only one round of division हो रहा है, and this is non-vulval fate. Okay, but the primary and secondary they have the vulval fates. Okay, and also if you see this pathway I've already mentioned previously, the LIN3 pathway. सो आई मेंशन आउट है कि कौन यहाँ पे इनहिबिटर है एंड कौन यहाँ पे रेगुलेटर है, कौन किसको एनकोड कर रहा है. सो यू कैन गो थ्रू इट. दिस जस्ट फॉर योर इंफॉर्मेशन. सो यू कैन सी. तो अगर यहाँ पे लीन 1 अगर इनिबिट हो जाएगा तो वल्वल फेट चेंज हो जाएगा. ओके, so these are all there. So I hope you have liked today's class. So I've already explained to you about why C. elegans is used as an experimental model, because एक तो है कि उनका life cycle बहुत short होता है, also वो intransparent होते हैं, so it enables researchers to observe in details. Okay, उसका genetic engine screening में it is very easy to use. Also rapidly it identifies functioning of many genes. और इसको हम कभी भी we can anytime we can freeze this C. elegans. Okay, it can be freeze and it can again be recovered आफ्टर लॉन्ग टर्म स्टोरेज, जो कि बहुत ज़्यादा इंपॉर्टेंट है. और आप लैब में काम कर रहे हो, द मॉडल शुड बी इजीली फ्रीज और इट कैन बी इजीली शुड बी इजीली रिकवर्ड. तो वो भी है. तो दैट्स व्हाई दिस सी एलिगेंस इज अ वेरी गुड मॉडल फॉर एक्सपेरिमेंट्स. ओके, so I hope you have liked this today's class. Do share it with your friends and subscribe to my channel for more such videos. Thank you so much. Take care.